Okay, ladies and gentlemen, here we are on part 2 of Unit 2. So here we go. History of cells. Obviously, you're studying microscopes. We are going to be looking at cells. So how cells were discovered. First of all, Robert Hooke was the first person to observe cells in court. He actually coined the term cells back in 1665. Cute little story was that when he saw the cells, it reminded him of the tiny little blocks that monks live in. So that's where the term came from. Um, about 150 years later, 160, Robert Brown discovered the nucleus. And then following that, about 5 years, we saw Schleiden, who theorized that all plants are made of cells. And then we had Schwann, who followed that a year later, saying that all animals were made of cells. And we'll look a little bit more about this in class at the history of cells. There are 3 components to the cell theory. The 1, all living matter is composed of 1 or more cells. 2, the cell is the structure and functional unit of all life. And 3, all cells come from pre-existing cells. Again, we'll talk more about this in class. There's a great song. I wouldn't say it's great, but you won't forget the cell theory. If we look at cells, there are 2 big groups. One is prokaryotes. And these guys lack a nucleus and other membrane-bound structures. So we can see prokaryotes right here. The other are the eukaryotes. These have a nucleus and other membrane-bound structures. So I always remember this by you carry a nucleus. Okay? Both types of cells have the following. They have cytosol, which is that semi-fluid material within the cell. They have a cell membrane. They have ribosomes, but these vary in size. And lastly, they have chromosomes. Now remember, a prokaryotic cell does not have a nucleus. But what it does have is a region of DNA, which we refer to as the nucleoid. And this is just that single strand of DNA chromosome that it contains. No nucleus, but it still has that chromosome. So for example here, if I show you this little drawing of bacteria, this diagram, here's my bacteria chromosome, and you can see here how it's also labeled as the nucleoid, or the region that contains the actual DNA. On the prokaryotic cell, you can also see, yes, it does have a plasma membrane. It also has a cell wall. And then we'll talk a little bit more about flagella when we get to microtubules later on. Okay, one of the things that I like about this picture is that it shows, oh hey, look, this is a eukaryotic cell. Look how big it is compared to a prokaryotic cell. A prokaryotic cell, the smallest prokaryotic cell is a type of bacteria that ranks 0.1 micrometers, very, very small. But most bacteria range from 1 to 10 micrometers, where eukaryotes are going to range from like 10 to 100 micrometers in size. So we see a huge difference. Why are cells so small? Well, it's for their advantage, meaning cell volume to surface area ratios favor small size. The nucleus to cytoplasm consideration, meaning we don't want our cytoplasm to be so big that the nucleus can't actively control it with the information it contains. Metabolic requirements, the larger the cell, the more energy it's going to need. So the smaller the cell, it's easier to sustain and regulate the materials that are going in and out, which brings us to speed of diffusion, which is the movement of substances across the cell membrane. So let's take a look at this image here. Yes, I stole this one from your book. Okay, what you are seeing here is the surface area to volume ratio. The smaller the cell, the higher the surface area to volume ratio. Meaning this, the smaller the cell, there is more surface area to support the inside of that cell. Okay? For example, in this one, if you look at the small guy here, letter A, he has a total surface area of 6. Height times width times number of sides times number of boxes. But a total volume of 1. Height times width times length. So his surface to volume ratio is 6:1. So he has a 6. Now check out the big guy. The big guy here is this one big cube. So it to me looks like a big UPS moving box. Its surface area is 150, where its total volume is 125. So now we're getting really darn close to a 1-to-1 ratio here. That's not good for a cell. We need to have the higher surface area to volume ratio, the better. And how do you get a higher surface area to volume ratio? The smaller the cell, the higher that surface area to volume ratio is going to be. Now here they showed you on this last one, oh, well, look what happens if we increase the surface area by putting indentations onto the cell. Okay, breaking it apart, making it look like, giving little divots so that it has more surface area. Then we're going to increase our surface area to volume ratio back to 6. So let's take a look at basic cell organization. Whoops, too fast. Okay, basic cell organization looks at the following: cell membrane, nucleus, cytoplasm, and organelles. We are going to talk more in depth about the cell membrane when we get to looking at transport of materials, and that's where we get to go back to phospholipids. Yay! But it's more in a biological sense, so don't freak out if you're sitting there going, oh, I hated that biochem. So let's take a look at what a typical animal cell looks like. In this typical animal cell, here you're going to see the plasma membrane, aka the cell membrane that surrounds the entirety of the cell. And you're going to see your nucleus. And then in here we see our cytoplasm. You're then also going to see several of these membrane-bound organelles, which we're going to spend a lot of time going in depth on, and a little bit more in depth than you did in Bio 1. In a plant cell, we're going to see the same thing. We're going to see that cell membrane again. We're going to see the nucleus. We're going to see the cytoplasm. And we are going to see membrane-bound organelles once again. And again, we are going to get much more in depth on this. So let's take a look at the membrane. And we're not going to spend, like I said, a lot of time on this right now because we're going to go more in depth later. But the membrane, the purpose, it's the protector, it's the gatekeeper. It is the guy who determines who gets to go in and who gets to come out. And his job is to separate the cell from the environment. It regulates the movement of materials in and out of the cell. So hence, that's why I like to call him the gatekeeper. The cytoplasm, or you might hear the term cytosol, oops, let me move myself, is the cell substance between the cell membrane and the nucleus. Okay, so our cytoplasm is, or cytosol, is the cell substance between the cell membrane and the nucleus. This is the fluid part of the cell. It exists in two forms. At some spots it's gel, it's very, very thick, and then other parts we see it more as a sol, which is a fluid. Organelles. The term organelle literally means little or small organ. This is a formed body or compartment in the cell with a specialized function. This is incredibly important in the organizational structure of cells. If you're thinking of a cell as a city or as a factory, these little subunits, these parts of the cell, are essential in the whole process of obtaining energy. Every single function in that cell is looked upon, is divided into these tiny little compartments called organelles. Oh, here we go. So it's a way to form compartments in cells to separate chemical reactions. What a huge, huge evolutionary advantage that we as eukaryotes, because bacteria do not have, prokaryotic cells do not have membrane-bound organelles. So we as eukaryotes have the ability to separate those chemical reactions so they don't interfere with one another. In addition to that, it keeps various enzymes separated in space so that they are not one competing with one another, let alone interfering with one another. So as we go through all these organelles, you must be able to identify the major organelles, give their structure, give their function as well. Okay, we get that? You must be able to identify them, give their structure, and give their function. So we're going to start off with the big dog, or what I like to call your mama. Your mama is the nucleus. And the nucleus is the most conspicuous organelle, meaning it is the most prominent organelle. It is usually spherical, but it can be lobed or irregular in shape. It's the one right here, right smack dab. And we often say in the middle, even though sometimes it's not in the middle. Its structure. It is surrounded by a nuclear membrane that is porous. You can see pores here in the nuclear membrane that regulate what goes in and out. It has a nucleolus as well, which is, oh, well, hold on. I'll tell you a little bit what the nucleolus is. I'm getting ahead of myself. Sorry. I love the cell and I get excited about it. Chromatin is the thin thread-like structure here, and it's DNA. So let's take a further look at the nuclear membrane. The nuclear membrane is a double membrane separated by only 20 to 40 nanometers of space. Okay, very small area here. The inner membrane is supported by a protein matrix, which gives the shape to the nucleus. Okay, so the structure, the proteins here determine the shape of the nucleus. It contains nuclear pores, I said poles, good gravy, pores that are regular holes through both membranes. These pores are only 100 nanometers in diameter. Remember, a nanometer is 10 to the negative 9th of a meter. We're talking ridiculously small here. Again, the protein complex gives shape to these pores and to the nuclear envelope. These pores are what allow materials in or out of the nucleus. So we're going to be pretty tight security here because the nucleus contains DNA and we want to have tight security. Now the nucleolus is the dark staining area in the nucleus. There are 0 to 4 nucleoli per nucleus that you will see. It is the storage area for ribosomes, and it is where ribosomes are made. And I apologize, I want to make sure you get that down. Okay, so storage area for ribosomes and where ribosomes are constructed. So here you can see some TEMs of what the nuclear pores look like. Okay, the nuclear lamina, which is the gaps or the spaces in between the, oh, it's not this. There, can you see that right there? Hold on. That is the nuclear lamina right there on the close-up of the nuclear envelope. Okay, where you can see your outer membrane, your inner membrane, and the lamina, which is the underlying tissue there. And there's our surface of the nuclear envelope. Okay? Love TEM. You can see some cool stuff, guys. Chromatin. Chromatin, chrome means colored, tin means thread. DNA and protein in a very loose format. And this is what's going to coil up and form the cell's chromosomes. We will get into big time detail on this with mitosis and meiosis. The function of the nucleus. Here's why I call it your mama. Because it's the control center for the cell. It directs everything. And it contains the genetic instructions for all functions of the cell. Everything. It controls it all. Just like your mom. Got it? Okay. We're going to end there. We're going to finish up with ribosomes on a second one since I'm getting close to my 15-minute cutoff time. All right. So get those down and we'll pick up and we'll see you later.